Okay. So welcome to PHAVES number 5. And I have our speaker who I can spotlight here today. So this is Evelien Adriaenssens from the Quadram Institute in the UK. And she is a career track group leader. And she's here to do an AMA. So she's a new PI. She works at a research institute, but she went through all the standard PhD and Phage stuff and then did a couple of postdocs. She's done a lot of moving for her postdocs to different continents. I know she was in South Africa and before that in Belgium and other places. And so she can talk a little bit about the career side, but also she is part of the International Committee on Taxonomy of Viruses. So she's the one who's approving if you say, I found a new taxa of a phage, she's part of that. And so she can talk about that too. And then the virome is kind of her area of research. And so you can feel free to ask her about phages that are in viromes, and bioinformatics is also kind of within her wheelhouse, the main part of her research. She's in the computer side of the phage world. She doesn't have to be in the lab right now, so ask her questions about that. And I know that her work right now is going toward gut virome stuff, so yeah, maybe you should ask about that. And I should say the format today is the first time we've done it, an AMA, so an Ask Me Anything. And so that means the entire time is just questions from the audience. There's no slideshow, there's just questions. So you can figure out what you can learn about Evelien today. And if we're gonna be checking the Slack and the chat of this Zoom room for your questions. So yeah, and then, so I'll let you kind of get started. People can ask questions, and I'm just gonna say a few announcements while we're letting the questions come in. So about some other things happening this week before I kind of turn it over. So we have today at noon, there's another phage event called the Quadram Institute for Physics is hosting a bacteriophage forum. I don't know if people have, they started it last week. So that's today at noon Pacific, so not too long after this ends. That's with Graham Hatfill and Joshua Weitz, and they're both going to be giving a talk, so that's going to be great. And then Phage Canada is on Friday, if you're part of Phage Canada's virtual series, that they have a session on Friday. Then in terms of conferences, Phage Oxford is coming up in September. You can register now. That is them. They normally host a conference in person, obviously, and it'll be online. And then the last one I wanted to mention is that we're starting this new collaboration with Viruses of Microbes team, they hosted the Portugal, or they were going to host the Portugal meeting this summer that sadly many of us wanted to go to, but we couldn't. And so they've switched it to next July, but they're gonna start doing webinars starting September 17th. And we're gonna be kind of their tech support hosting partner. So the first set will be, the speakers are Rob Edwards, Ruth Ann Sanda, and Colin Hill. So come in for that. And all of these are posted in Capsid and Tail. So you can check for details and registration links. Okay, so we have a first question. Thanks for staying tuned. It's from Jan. I have a first question. The ICTV seems to have been really productive in creating new taxa. What's that like getting buy-in from NCBI, and have you been getting a lot of pushback from other researchers? Go for it, Evelien.
Wow, this is like immediately a full-on question. Thank you, Jan.
Sorry.
No problem. So firstly so we've been indeed creating a lot of new taxa. One of the reasons why we're doing that is because before everything was unclassified. And a lot of, if you had a phage genome, the chances were very high that it would be unclassified type of Virulent, for example. So first thing that we started to do is we're making new genera based on genomic similarities so that we could at least have a genus and a species affiliation for most of the phages. So we get a lot of buy-in from NCBI, and even and NCBI even says they will not classify anything unless we give the go-ahead. So because the ICTV is the mandated committee by the International Union of Microbiological Societies, NCBI has decided that they will only listen to what we say. So if I, for example, say I cannot classify this, then it will remain unclassified. Now, what NCBI does is it follows the general taxa that we give it. And if they get new genomes that are submitted, they will try and see whether it already fits in something that exists, and then they will just put it there as unclassified this genus, or unclassified this subfamily, or unclassified, until we formally state, okay, it will go into this bin. So getting pushback from other researchers, I think in general, people see phage groups, and what we try to do is give them a name. So we generally agree with everybody about this is a group of phages. We do phylogenetics or comparative genomics, and everybody sees this group. And what happens is that some people say this group is a subfamily, some people say this group is a genus, and some people say this group is a species. And it depends on what kind of background they have. So for example, a lot of the dairy phage people have been used where phages are something you don't want, and who have a lot of contacts with companies. They will say, oh, this whole group, we see this as a species because it's easier to communicate with companies. Whereas for myself and a lot of other people come from the phage therapy perspective, where you want each isolate almost to be a separate species, because then you can say we have so many different species that we combine into a cocktail. We we had to kind of make a decision on where the thresholds lie, and they are in favor of more the phage therapy part than the dairy phage part. But that's also because there's a lot of metagenomics that's being done, including by me, and you need to have genome-based boundaries for that. Otherwise, all of your sequence reads will come back from your sequencing as unclassified. So I hope that was a quick response to your massive question.
Thank you, that's really good.
Awesome. Yeah, starting off strong, trying to find controversy. No.
Yeah.
All right, so here's another one. Hello from Edinburgh, Scotland. This is Gonzalo Yebra. Evelien, what would be your recommendation for someone who finds a prophage or phage in a bacterial genome? How do you know if it's unique? Would you recommend uploading it to Genbank? How would you name and classify it? There's another cluster of questions.
Yeah, so when it comes to prophages, they're not my area of expertise, but we've been getting more and more questions about them. And there's one important thing at the moment that we need before we can classify them is we currently only classify them if they're confirmed as virulent, so if they're confirmed active. So that you can clearly know what the phage genome is within the bacterial genome. So one of the things that the taxonomy committee needs is a genome sequence for its records. So if the only genome sequence that is available is the bacterial genome sequence with coordinates from here to here, we currently cannot really classify that. But in the future, when people try to verify their phages, their prophages, or whether they can submit to Genbank as a third-party annotation only the phage genome sequence, and then it will be possible to classify it. But this is kind of contentious, and I cannot just give a general answer to it, because we don't want part of bacterial genomes to suddenly be in the virus reference databases based on predictions that we're not sure of.
Awesome. Okay, we'll go to the next question. Thank you for that one. Okay, Namika Nadi says, for a lab that does not have capacity to do genomics, How can one tell if a phage is virulent or not without sequencing?
Ooh, that's a good question. So, and I haven't been in a lab for a while myself. So maybe I should pass this on to my postdoc, but no. What people generally do is check whether the plaques that you see are turbid or not. And it's generally accepted that if your plaque is very clear, that it is a virulent phage, and if it's turbid, that it has the possibility of being a lysogenic temperate phage. But there's a lot more to it than that. So it would take probably lysogenic conversion or lysogenization experiments to verify that your phage cannot integrate in the genome. So maybe something PCR based. I haven't actually thought about this in detail, so I need to do some conceptual thinking about this.
Thank you. Yeah. Yeah, okay. Ebenezer Foster Niarco says, how would you explain what a phage is to a 9-year-old asking for my daughter?
Okay. Eben's from my institute, so thank you for this question, Eben. I do not have any children, so I'm gonna try and put myself in the thoughts of a 9-year-old. So a bacteriophage is a little killer of bacteria. So kids can get sick, but not only kids can get sick, bacteria can also get sick. And when they get sick, they get a bacteriophage. And we can use the bacteriophage to kill the bacteria. So the enemy of your enemy is your friend.
Ooh, I like it. Any 9-year-olds on the call?
Well, it's a good thing because I have a nephew who's 8, so I need to start thinking about this for when he starts asking me questions. What his auntie does.
Yeah. Okay, Atif Khan, I have started my phage research 2 years back and optimized some basic protocols. According to you, what are the areas where I should start investigating?
Whew, that's also quite a big question. I think there's loads of hosts where we don't have phages for, and that's definitely an area to look into. I think there's loads of phage types that we only know from a couple of model hosts like E. coli. For example, there's only a couple of lipid-containing hosts, phages. There's only a couple of RNA phages known in isolation. So obviously there's reasons why, because it's not as easy. So it all depends on what your bigger research goal is. Do you want to know more about what phages do in general? Do you want to know more about phage diversity? If you want to know more about phage diversity, it's definitely looking at the hosts that aren't very well known. Or looking at environments where we haven't looked at for phages before, which is one of the things that I did in my postdoc, is we're looking in Antarctica and deserts and the Namib Desert to see what the viral communities were like in very, very dry soils. And we did indeed find phages, and some of the phages we did find, and this is with culture independent approaches, they are actually quite similar to known phages. So sometimes the world is small and sometimes the world is big.
Wow. Are the phages in the Antarctic samples like very cold stable? Or I guess every phage might be more cold stable.
Yeah, so it was really cool because we had data set, we had soil. So basically we went to scoop sand, and I didn't go to the Antarctic myself, my boss did, but I did go to the Namib Desert to scoop and to scoop sand and to look at it. Okay, it's a bit making it sound too easy, but so we had a hot desert and a cold desert to compare, and
Wow.
there's definitely a fridge/freezer effect going on in the Antarctic, where you see loads more particles conserved in the Antarctic than in the Namib Desert, and probably also according to water availability, areas that are drier and wetter, you see more a trend towards more lysogeny when it's drier and hotter, and more extracellular particles when there's a little bit more water and when it's really cold. So that's probably also because, you know, the particles have been conserved for who knows how long. We haven't been able to do any carbon dating on it, but
Wow.
it might have been thousands of years.
That's insane. Okay, we're going to switch to a career angle. I recently started phage work. We're beginning to look at phage antibiotic synergy. It's been almost impossible to get a postdoc. What will be your advice for a career progression to get grants and build a formidable phage lab?
So I can't really see the question at the moment. Are they asking for they themselves being a postdoc or hiring a postdoc?
I think, oh, that is a good clarifying question. I don't think I can make that assumption, actually. We'll let Namika answer that if you're still listening, but maybe you can pick one side of that and comment.
Okay, so finding a postdoc, in my personal experience, is as in, if you want to yourself become a postdoc, has been kind of almost a cyclic nature to it. Sometimes there's a lot of positions available, but each time I'm looking, there's none. Or at least that was my feeling of it, and I think a lot of people get the same feeling. Each time you're not looking, suddenly you see all of these adverts for these amazing positions, and you're like, "Where was this 2 years ago when I was looking?" So a lot of it is luck.
Oh, we have a clarifying comment.
Thank you so much.
Or is that just?
I think there should,
Echo from.
Okay,
just keep going.
Addison, come here.
I think somebody's calling for their kid. So if you're looking for a postdoc yourself, for me, a lot of it has been luck, seeing a position advertised that I didn't, in a lab that I didn't know of, and then just going for it. And another part of it is meeting people and talking to them and building your network within the phage community, and then being informed by people when they have a position. So my career progress has been a combination of both. And yeah, as always, luck is a big factor in my opinion.
Yeah. How about the flip side of that? Because you had to hire a postdoc recently, what was it like on the other side?
It was very weird because I did not know what to expect. And my biggest fear is that nobody would want to work for me. Especially because I was new, moving to a new institute. The institute itself was not that well known. So I kind of tried to make, try to advertise on Twitter and asked my colleague PIs to advertise, and I had loads of applicants. So I don't know if that was part of the fact that we were in a situation where there was more people looking for postdocs than there were any available, or that people for some reason want to work with me. So I was very lucky that I that had a lot of choice and was able to find somebody who really fit what I was looking for in the lab.
Awesome. Okay, let's go to the next question. We've got lots of questions. Thank you guys all so much. So Clement Cocle, that's probably said wrong too. Hello from Pretoria, Cowan's lab. What do you think about working on virus communities from viromics or from metagenomics data sets? What are the benefits and disadvantages of these two methods?
Okay, so I'm guessing what the question is in the sample prep, you look for virus-like particles and only sequence that, versus you just sequence everything and you have a metagenome data set and then you go look for phages. So both of them have their advantages and disadvantages, sorry. And so what I've been trying to do recently in my most recent set of viral-like particle extracts is I've been looking for both the DNA and the RNA viruses. And that's something you can't do when you just do general DNA metagenomics. And I've been very lucky, hopefully I need to submit this paper today or tomorrow. It will be published soon, but when you do that, you can have a lot of RNA phages that you find, which get underreported in all other data sets. On the other hand, if you look at the full metagenomes, you can also find obviously all the prophages and the prophage potential that's in the metagenome data sets. But because you invest a lot of sequencing yield into sequencing the full bacterial genomes and maybe microbial eukaryotes and stuff that are in your sample, you need to do really deep sequencing to get to the same quantity of phages and viruses that you have in your viral-like particle extracts. So if you're only looking for phages or viruses in your sample, I would suggest to do the viral-like particle extracts, because otherwise you're just going to pay through your nose for the sequencing and the bioinformatics afterwards to pull it all together. So it basically depends on your research question and your budget as the two, I would say, the two main criteria.
Thank you. Awesome. Interesting. Okay, from Madhav. Hello, Dr. Evelien. Greetings from India. I'm a little bit confused on the Myoviridae and Cypoviridae classification. Can you enlighten us on some of the quite specific features for distinguishing them? In case where sequencing is not available, can we distinguish based on tail size or other features based on TEM morphology?
Okay. So this all comes down to the way phage classification used to be and the way we're trying to take it forward into the age of sequencing. So the way phage classification used to be is that it was based on the morphology and the content of the nucleic acid. So the majority of phages, and this is still the case, are double-stranded DNA phages with a tailed structure. So then based on the tail structure, whether it's a short tail, and then it was classified in the family Podoviridae. If it had a long non-contractile tail, it was classified in the family Cypoviridae. And if it had a long contractile tail, it was classified in the family Myoviridae. And these three families were then grouped in the order Caudoviralis. Now, when you make a tree, looking at any type of features of the genome, or any clustering of the genomes of these three families, they don't come in three groups. And that one of the reasons is that the genome is not necessarily always a good predictor of the morphology, and vice versa. You have groups of phages, lambda-like and P22-like phages that are in two different morphological families, but have shared genome characteristics. But at the same time, you have other phages that are classified in the same morphological family that have no sequence identity. And you have phages that are over 200 base pairs in their genome in the same family as phages that are only 70,000 base pairs. So what we're trying to do in our phage classification of the future is pick those groups apart and make monophyletic groups that are new families within that. So unfortunately, that means that you need to do sequencing to classify your phage. There's no way around it. There are no morphological features that we can use to consistently predict the genome-based families and anything below.
Wow, thank you. Okay, from Henny, check the growing colonies and, oh, maybe I missed some part of it. Check the growing colonies and perform an induction experiment. Compare with wild type. Oh, this was in response to one of the questions, I think, for whether you had a lysogen, was that?
Yeah. So I think what Ani is kind of trying to say is that you can also check whether your host strain has a prophage in it by trying to induce it. And compared to the wild type. I was thinking of the question, if you isolate a phage from a prep, how can you say whether it integrates? Because that would be a new prophage possibly for your phage. So there's a lot of things to think about.
Awesome. All right, we've got some ICTV, another question there from Adriana. What is the ICTV guideline identity threshold to classify two phages as two different phages? Are the guidelines the same for metaviromes?
Okay.
Hi, Evelien.
Hi, Adriana. Nice to hear you.
Save that question for RNA phages.
Okay, so there's a semantics issue at work here as well, because there's a difference between a phage and a phage isolate and a phage species. And the best way to illustrate this is saying that there's 7 billion isolates of the Homo sapiens species, and they're all different. In the same way, a phage species, it's just a box or a bin that we put isolates in, and each species has an exemplar, which is kind of the type. If I was to say Jessica Sasher is now the exemplar isolate of all Homo sapiens.
Wow.
This way, this way, all phage species have their exemplar. So then you can compare exemplar or isolates belonging to a species to others. They belong to two different species if their genomes are more than 5% different. And that's just a short threshold, genomic-based threshold that we use, which was chosen kind of abstractly based on the available information that we had at the time. It seemed like a good idea. If somebody, and I'm now looking at Andy Millard somewhere, has a better threshold that they want to impose, they should publish their data saying that this is the better threshold. So in that sense, if two phages are different in one SNP, I can say that they're two different phages, but they will be classified into the same species. And that doesn't, and so what we try to do is for each of the taxa that we make is to define the thresholds and to write down the thresholds so that you know whether something you have is new or not. So for the metaviromes, the guidelines are slightly different. So we did a meta-analysis that was done by Simon Roux from the Joint Genome Institute 2 years ago, and that was published as a paper called MyUVIC, so minimum information for uncultivated virus genomes. And there we basically combine a couple of the species level thresholds into one, taking into account that your contigs that you assemble from a metagenome might be different in length. So the threshold that we made there was anything that's 95% similar or 85% of the contig length, comes into one viral OTU, so viral operational taxonomic unit, that you can put equivalent as a species, but it's not. It's a broader group than a species. But for the sake of metaviromes and viromes, it works quite well.
Great. Yes. We wrote a capsid and tail article on that paper long ago. I don't even know what I said, so don't go back and read it. Or do it.
I know it's Jan's favorite paper now.
Yeah, Jan loves this stuff.
Yeah, just as a note, I've been building a lot of like code that verifies schema, and that's what we're doing to build our own databases. So like I already have like privately a thing that verifies MyoVig in terms of like the schema that we're going to have people enter in, but that hasn't really been released yet because it's really buggy, but eventually it will be released and everyone's going to be able to access it.
It was all born out of that work.
It's all Simon.
Adriana, were you
Can I build up on that question?
Sorry?
Can I build up on that question?
Go for it.
Yes.
So, in that sense,
can I cancel the spotlight? I want to look at
Oh,
Adriana.
yeah. Sorry.
Yeah. All right. Yes. Now I see you.
So, in that same sense, what was the threshold for you to consider two phages as being the same phage?
It's still a conceptual issue, and I would say one SNP difference. You can call it two strains of two different isolates.
That is. Are you satisfied, Adriana?
Yes, thank you.
Okay.
Thanks, Evelien. Nice to see you.
Okay, Juan Martinez. Hello, greetings from Mexico. I have a question about phylogeny. While some people use the amino acid sequence of the DNA polymerase or capsid with tools like Victor, what do you recommend?
So what we're trying to do with our genome-based reassessment of the whole viral world, not ambitious at all, is also evaluate some of the tools, right? And my conclusion is that you can't beforehand predict what tool works best or what marker gene works best. So depending on how broad you want to look, do you want to look at something that's a whole group that's very distantly related? Then you'll have to go for methods like the phage proteomic tree, which you can now access through the VipTree server online, which kind of compares the whole predicted proteome of your phages with the whole predicted proteome of all other phages. The same can be done by vContact, which then generates a network and predicts clusters of phages that belong together, that have some sort of taxonomic affiliation with each other. So what we've done recently is we defined a new family called Hirelviridae, and then we looked at what kind of tools you can use to define the internal structure. So for example, for this group of phages, they all had introns in some of their conserved core genes that you would normally use to build a tree. So we didn't want to use, for example, the terminase large subunit to build a tree because we couldn't 100% certain identify the boundaries of the intron and the actual gene. So if there's mistakes in there, that will carry through in your phylogeny. So we looked for other genes that were shared among the whole group. So we basically did a core genome analysis as they do it in bacteria, in bacterial Genomics. So you define a set of orthologs that are present in all of the phages that you're looking at, and then looked at single marker gene phylogenies and concatenated protein phylogenies of all of the core genes together. So, so I can't predict beforehand what works best for your group of phages. I can say that a capsid is a safe bet, a terminase large subunit is a safe bet. But there's other things to look at, especially when you have certain groups of phages that recombine modules, so that you have mosaicism, which is not all groups of phages that do that. So it will depend on that.
Great. Okay, we were switching back to careers. Did I understand, this is from Jolene, did I understand correctly that you did multiple postdocs before starting your own research group? How did you decide when you were ready to start on your own and when it was better to try a different postdoc?
Oh, yeah, that's actually a good question, because for me, so I'm not one of those people who had their whole career planned out when they went to university. I kind of organically rolled into a lot of the positions that I found myself in. For example, when I was studying, and I studied at the University of Leuven in Belgium, which was immediately kind of a 5-year master's degree. We didn't have the distinction between undergrad and postgrad. So I always said, I'm never going to do a PhD. I never want to do a PhD. One of the reasons why I said that is because I'm a first-generation university student, and I didn't know what that entailed, a PhD, up until I went to do my master thesis, and I was in a research lab, and I suddenly realized, oh, I'm actually good at this, and I like it. So then I did a PhD. So in the final stages of my PhD, again, the question arose, what am I going to do? Because in the beginning I said, I'm never going to do a postdoc because I'll get a real job. And then I was like, okay, the whole postdoc thing, it appeals to me. I think I'll be good at it. So I'll do one to try out. So I got a grant for 2 years to go to South Africa to join the lab of Don Cowan there. And I liked it. And again, it turns out I liked it and I was good at it. So I decided to get another fellowship to stay in that lab for a second postdoc. And then the question was already in my third or my fourth year of doing a postdoc, what am I going to do? Because then I was looking at the different countries that I might want to live in, and I was looking back at Belgium, and it turns out that I was already too old and too senior to apply for any of the postdoc positions there, because they generally only allow you to be a postdoc for 6 years in 2 rounds of 3. So by the time I was applying, the 3-year cutoff had already come and gone. So that was kind of out. So then that made me realize, okay, this is kind of the point where I have to make a decision. Do I want to apply for, in Belgium, it would be professorships, in the UK, it's lectureships. So I tried a bit, but then I saw there was a postdoc that was advertised at the University of Liverpool by Heather Allison, who I had met at the Evergreen Phage meeting in 2011. So that was way before I met her. Like 5 years later, I was like, hey, Heather, you have this really cool postdoc. I'm really interested. And then I applied for it and I got it. So that's when I moved to the UK. And only then I started getting serious in moving towards a PI position. Because at one point, I was over doing a postdoc. In the beginning, I was very happy that I didn't have to apply for grants, that I didn't have to be responsible for anything. And then gradually it kind of organically grew that I wanted to have more responsibilities, but I wasn't allowed to. I wanted to supervise a PhD student, but I wasn't formally allowed to, even though I did in practice. And all of these points, and at one point you say, okay, this means I'm ready. And some people aren't, and some people are. And then I made the decision to not apply for any more postdocs and to only apply for fellowships and for lectureship positions and PI positions. And that's how I ended up where I am now. But it was a stressful time, because at one point I thought, oh no, do I have to like move into my parents' attic if I don't find a position in the next 6 months? And I was like seriously considering doing that. But.
Awesome. Jolene is smiling, so I feel like you gave her a lot of enlightening advice.
Yes, definitely. You said a lot of stuff that was like very relatable. So it makes me feel like okay about feeling how I'm feeling with wondering where my career is going. And so I'll trust that I will feel it eventually when I'm supposed to go somewhere else. So thank you so much.
Yeah, and I have colleagues who were postdocs who kind of never felt that. And they were then actively looking for a staff scientist position to stay, because postdoc is the level that they were comfortable at and wanted to do. And there's no right or wrong decision unless you feel uncomfortable with one of your decisions.
That's really good advice. Thank you.
Thank you. Okay. So we don't have a ton of time for all these great questions because we're going to still make sure we have room for at least one breakout room, but I think we'll ask a few more. I'm looking at just to see if there's any that cover something that you haven't hit on. Oh, there's lots of awesome questions. I think we should put the ones we don't get to in the Slack, and maybe you can get to them at some point. Okay, let's just go to the next one. From Juan Martinez, another question about viromics. Do you recommend the use of tools like ResFinder or Resistomed gene identifier to detect resistance genes, even knowing that these tools are trained with bacterial genomes?
I've actually started trying ResFinder on some of my data sets. And one of the things that I found out by using ResFinder is that, my viromics data set had mispredicted a lot of plasmids as being viral. So one of the things to consider is that antimicrobial resistance genes are very, very uncommon in phage genomes itself, in the phage genome themselves. That doesn't mean that they cannot be adjacent to the phage genome and get mobilized that way, but on the phage genome itself, there's only, I think, like 2 or 3 verified instances that it's there. So whenever you use a tool like ResFinder to predict antimicrobial resistance genes, and your prediction says there's an antimicrobial resistance gene on a phage genome, I would always be very wary of that. And first of all, check whether it's actually the phage genome or adjacent to a phage genome on a prophage, or whether it's one of those commonly misidentified genes that are not actually conveying antimicrobial resistance. So the next step is to actually test whether it is antimicrobial resistant. And I think Marie-Agnès Petit from INRA in France has done quite a bit of research on this, and I consider her an authority on this subject. And she has done extensive research on viromics and AMR, and she said there's usually no AMR genes on the phages in the viromes. Wow. And a lot of the reported ones are actually misreported.
Wow. Okay, let's go to another one. Adriana, phages with similar genomes are currently part of different families, example, lambdoid phages, because although they have different morphology, they have a similar genomic structure. Is ICTV contemplating this issue?
Yes, we are contemplating the issue, and we have neglected it for a long time. So in March, I asked Susan Lamond to be the chair of a newly formed lambda-like virus study group within the ICTV bacterial virus subcommittee, and she has started looking into it. She's working together with, again, Marie-Agnès Petit, and they have a little working group together where they're looking at how to tackle the problem that is lambda with its different morphologies and a lot of recombination and module exchange. So if you are interested in this effort, Adriana, I can always give your contact information to Susan, who would be happy to have you helping out.
Awesome. Well, Adriana's smiling, so I think that means yes. Okay, and we're going to do a couple more questions, then we're going to do a breakout room, and then we'll get back together just at the end of the hour to kind of close out, and maybe you'll have a bonus session at the end. People want a second breakout room, but I don't want to miss too many of these questions because it's been great. So from Ritam, what's that?
I said, let me sip from my
yes,
water.
take your evergreen water drink. So Ritam Das says, hello, Dr. Evelien. Wanted to know. Oh, I hear Betty.
Hi, Betty.
Hi there, everybody.
Wanted to know if there's any database of phage mutants. Do we need to submit whole genome sequence of mutants generated in the lab?
That's a good question. There's definitely not in the big data bases like so NCBI, EBI, database of Japan. I don't think there's a separate phage mutant section. So if you want to submit a whole genome sequences of that differ only in a couple of SNPs or in indels from other sequenced genomes, that's not necessarily an issue. You can just submit them as normal genomes to Genbank or like any of the INSDC databases. What will happen is they'll just all belong to the same species. So the only known thing where I know that it happened, where there was a set of phages where the genomes have been in the databases for over a decade, and the description in the paper said they're all Bacillus anthracis phages that come from the same experiment, and they are descendants of the same phage. And then we did classify them together way back when, I think it was 2012 or 2013 or something. When I just started out doing taxonomy. So what we do have is, so NCBI uses our newly proposed exemplar genomes of phage species and they're starting to add to the RefSeq database. So normally only the exemplar phage genome of each species will become a RefSeq, get a RefSeq accession number and come in the RefSeq database, and all of the other isolates that are suggested to belong to an existing species will come into the NCBI, what they call their neighbors database. So they will not be part of the main RefSeq, but they will be part of the non-redundant nucleotide database.
Wow. Okay, well, let's do one last question before our breakout. So this is from Andrew Millard. Given the Myoviridae, Podoviridae, Cyphoviridae not monophyletic and the new families have/are being created, taking phages out of these families, are these going to be completely removed? ...
Yes, well, that's the goal. In practice, until we have completely divided up all of the different groups, we'll probably keep the Podomyocypho as a kind of rest bin for people to deposit their genomes into NCBI. Otherwise, all of the new genomes will come as unclassified Cadaveralis. And now they're unclassified Myopodocypho. So probably for the coming 2 years, they will still exist. And after that, it depends on how productive we are as a taxonomy committee, to which Andrew Millard also belongs.
Is the unclassified Cadaveralis actually more useful though than unclassified myoviruses, which doesn't really mean anything?
The difference is kind of, it's the difference between usually we've done TEM and we haven't done TEM.
Okay.
But from the genome perspective, I don't really see a a reason to keep the myopodocipho. From a practical perspective, abolishing it is a bit of work that somebody needs to do.
Yeah, it's that big spreadsheet that I can appreciate that.
Great. Well, thank you all for all these amazing questions. We're going to go to breakout rooms. If you know that you're not staying for the breakout room, this is the time for you to you can say goodbye now. And then I'll see the accurate number that I can divide up. So there's a few people leaving. While people are making up their mind, I want to mention that one more announcement, that Jan and I are looking to talk to phage hunters, especially people who run labs that have students looking for phages on the regular, or they're getting into it and might want to make some money from licensing non-exclusively these phages to companies that are trying to develop them into therapeutics and agricultural additives. So if you have phages and you're looking for them, talk to me. I would love to just ask you about this. If you think this is a terrible idea, come talk to me and tell me why. I would love to know that too. So with that, we're going to create the breakout room. Funneling back in. Hello.
Hello.
Awesome. Hi. So I hope you had fun. I'm just going to formally close the session. You don't have to leave if you want to stay for the bonus room, but people have their hour slot booked. And so we like to let people have an easy exit. So thank you so much for coming. Thank you, Evelien, for taking the time to answer everybody's questions. We'll be passing on the chat to her so that she can see the other questions. And if she wants to answer them in Slack, we'll be doing that. So you can join our Slack community.
Yeah, and just a reminder, we're looking for people who are interested in working with our tech transfer offices because we are trying to create a way to make it easy for you to share your phages, maybe sell your phages and so on. So we would love to talk with your tech transfer offices and build up these tech transfer to tech transfer relationships. Yeah, so we want to streamline it for them to make your lives easier as researchers.
Yeah, because we hear a lot about people who say I would love to share phages, but my tech transfer office or my university might have something to say about that. And you get the sense that they're the boogeyman that says no. And we're looking for the friendly tech transfer offices, but we're also looking for the boogeyman ones. And we want to talk to whoever your contact is at those universities so we can help create this system that makes it easy for phage exchange to happen. That's for research, therapy, and for companies. So thanks, for jumping in.
Are you willing to chat with government people?
Absolutely. Yes. Let's talk to them first.
That's so hard to talk to.
Just gonna give you a hard time. So I'm gonna put you with someone. You asked for it, so
yeah, that'd be amazing.
Yeah, we'll get in touch after this. Thank you, Hani.
Yeah.
Well, one thing in addition you may well be aware of or not. In general, the small colleges aren't doing a great deal of tech transfer and dependent on them. And in most of the colleges I know, there's no problem in terms of tech transfer. They're glad to get support from outside for any kind of research. And that's a great place to have students have projects where they're isolating lots of new phages. So the combination of those kinds of things could well be a way to get some additional money for doing this at colleges in all parts of the country. And
Thank you, Betty.
It's a wonderful way for students to be drawn into the phage field. As you know, there are a number now that were in my lab years ago, but
yeah, that's the gold standard. And that's what we've been hearing. So it's good to hear that feedback that undergrad student-led labs might be a lot easier to work with on the tech transfer side, but also they might get a lot more out of it and might be most excited to be a part of this. So we're kind of seeing them as bucket number one to kind of start talking to.
I think that's great. And I'll be glad to talk with anyone you like about experiences I've had.
Thank you. Yeah, we should create another meetup maybe to just talk about this for those who are interested.